Everyone. This is Dr. Bishal Gewali from Queen's University, Kingston, Canada. And it's my big pleasure and honor today to be speaking with you all virtually on long-term follow-up on single-arm registration studies, what have we learned, at the ESMO MCBS session at the 2022 Annual Congress of ESMO in Paris. I hope you all are having a lovely time in Paris. And I wish I was there in person. Unfortunately, due to passport issues, I could not make it in time. But I hope we'll have a fruitful and meaningful discussion, and I'll be available for live communication online after this talk. These are my disclosures. So first, let's discuss what single-arm registration trials mean. And in fact, is it even an oxymoron to say single-arm registration trials? Because these phrases, single-arm trials and registration, are not supposed to go together. Because traditionally, the purpose of single-arm trials is not registration or approval. The single-arm trials, phase 1, phase 2 trials, their purpose is to decide whether or not to take the drug to a phase 3 RCT. In fact, we did a study of a cohort of negative phase 3 randomized trials in oncology, and we found that 42% of them had no phase 2 and 28% of them had a negative phase 2. So that means we had done a randomized trial despite having a negative single-arm phase 2 trial, and we ended up with a negative phase 3. So traditionally, the purpose of a single-arm trial is to give us an idea about the activity of the drug. Is it an active drug? Does it produce any tumor response? The purpose of a single-arm trial is not to prove efficacy, which is proven through a randomized trial. So in other words, can we do approval or registration of a new drug based on single-arm trials alone, which usually is not the case, but things seem to be changing in recent years, as we'll see. However, in recent years, people are arguing that single-arm trials in and of themselves may be acceptable for approval if all of the following conditions are met. RCTs are not possible because randomization is unethical, which is rarely the case, because for randomization to be unethical, the drug should already be proven, which is not proven without an RCT, or because the disease is rare. And the safety signal is established and benefits outweigh risks, and the duration of response and the response rate is really high in a well-defined population, and the mechanism of action is strong, and there is an unmet medical need. However, all of these criteria do not have a clear definition, as we'll see. However, the question is, what is the right endpoint for single-arm trials then? Obviously, the time to event endpoints like PFS or OS that we use in randomized trials are not good endpoints for single-arm trials. This might sound a little counterintuitive because we always say overall survival is the gold standard endpoint, but for single-arm trial without a comparator arm, it is not, because it can be biased, because this survival can become a function of the natural history of disease itself, and it may not have anything to do with the intervention. But response rate is assumed mostly to be a function of the drug. Without the drug, why would there be a response? Because we know that the spontaneous regression of tumors, the shrinking of tumors, or complete disappearance of tumor without any treatment is exceedingly rare. So if there is a tumor response, that has to be a function of the drug, a function of the intervention. However, we need to keep in mind that the overall response rate of placebo is not exactly 0% though. There have been some studies in the past. One was done by Tanak in 2003 in Journal of NCI, where the response rate of placebo was shown to be around 2%. And there was one, a little more recent paper in 2015 that's also showed the response rate to be in the range of 2%. So the overall response rate of placebo is not exactly 0% though. Why? This is very intriguing, and this is subject of further research. And we are also working on a paper to establish the overall response rate of placebo in the modern era. However, it's not exactly 0% though. There is some response that is seen in randomized trials through placebo alone. So going back to our question of can we use single-arm trials for registration? And unfortunately, I think that ship has already sailed, because we already have a plethora of drugs that have been approved based on single-arm trials alone in oncology. But you might reasonably think that these are only accelerated approval, right? Because accelerated or conditional approvals can be given based on response rates, based on early sign of promising activity that has to be confirmed by a randomized trial in the future, and if it does not work out, it will be withdrawn. So it is natural to think that the single-arm trial-based approvals are conditional approvals. However, you would be mistaken. Because in modern times, we have seen several cancer drugs approved on the basis of single-arm trials, and they are receiving regular approval, not accelerated or conditional, but regular approval, which is surprising because the regulators seem to have acknowledged the pitfalls of single-arm studies. And for example, in this guidance from the FDA, they specifically mention single-arm studies for accelerated approval, and that is also only when there is no other available therapy and where major tumor regressions can be expected. However, in one of the recent papers that we did, we see that the number of cancer drugs approved on the basis of surrogates has skyrocketed in modern times, and a major percentage of that is response rate. But at the same time, the percentage of drugs that are approved on the basis of accelerated pathway are actually decreasing. That means more and more surrogate-based approvals are regular approvals instead of accelerated approvals. This was a very important and quite comprehensive analysis of the last 5 years of solid tumor approvals by the FDA, done by Professor Nathan Czerny. And here he shows that 58 of 161 drug approvals in last 5 years were based on single-arm trials. And of those 58, 39 got accelerated, but 19 of them were in fact regular full approval based on response rate from single-arm trials alone. Of those drugs that received accelerated approval, 4 were withdrawn, 8 converted to regular approval, and the rest continued as accelerated approval. But more interestingly, when he looked at the overall response rate for drugs that received accelerated approval versus regular approval, it was not different. It was a median of 40% in either case. So response rate magnitude alone does not explain why some drugs get accelerated approval and some get regular approval based on single-arm trials alone. So the ESMO MCBS scale has also recognized that more and more drugs are getting approved on the basis of single-arm trials, so they need to be scored for value assessment. But they clearly mention single-arm studies in the context of orphan disease and high unmet need, and they provide a grading from 1 to 3 based on single-arm trials. So the maximum possible preliminary score is 3 if the PFS is more than 6 months, or the response rate is more than 60%, or the response rate is between 20 to 60, but the duration is more than 9 months. However, after that, there can be some adjustment of downgrading based on toxicities, or upgrading by 1 points based on quality of life, or if there is a confirmatory adequately sized phase 4 experience, so that the maximum possible grade based on single-arm trial is that of 4. So going back to that analysis by Dr. Czerny on single-arm trial-based cancer drug approvals by the FDA in last 5 years, we see that the MCBS scores for the majority of them were 3, and it was just 1 or 2 for 20 of the 58 drugs. This is the lowest possible value that you can get. And although theoretically a maximum possible of 4 was possible, none of them had a MCBS of 4. And interestingly, all the drugs that had to be withdrawn later had an overall response rate of less than 20% and an MCBS score of only 1. So just as a case study, we can look at the drugs that are approved in non-small cell lung cancer based on single-arm trials alone, based on response rate and duration of response. We can see that almost all of them are biomarker-based drugs. Based on specific biomarker targets, and we can see that some of them have received accelerated approval, some of them have received regular approval, and the response rate is different, but it ranges in the range of somewhere from 30 to 60%. And the MCBS score is 3 for all of them, except the last one, which has a score of 2. And the duration of response is also varied. But what is interesting here is that if you look at this list carefully, you don't see any particular feature that differentiates drugs that received accelerated approval versus drugs that received regular approval. The response rate is in the similar range, the duration of response is in the similar range. So just by looking at the response rate and duration of response, you would not be able to say which drug would receive regular approval and which would receive accelerated approval. So why are we so much concerned about single-arm trial-based approvals? Because there are several issues that need to be addressed. The first is about the selection of the patients. In single-arm trials, the patients are highly selected, super fit patients, and because there is no concurrent control arm, we cannot adjust for known and unknown confounders. And then there is the issue of sample size. How small is too small? How big is big? The same is true for response rate and duration of response. Is 15% response rate in 50 patients good enough? Do we need 30% response rate in 500 patients? How do we define those boundaries? And as we discussed earlier, how to define what constitutes good enough evidence for regular approval versus accelerated approval? And how do we actually define unmet medical need? And then there is the issue of safety, which cannot be ignored because safety cannot be assessed adequately in a small sample size. And we'll be discussing these issues individually. So let's discuss about these points individually. The first point was about selection bias. As we know, single-arm trials, they have no concurrent control, and they must rely on historical control and presume that whatever response that you are seeing is a function of the drug and is better or worse than what we knew from the past experience. However, this interesting analysis that looked at the drugs that the FDA approved based on response rate, they looked at what would have been the best available response from currently existing drug. If you did not have that new drug, what would have been the response with the best possible existing drug? And they find that in several cases, as you can see, the response rate of the precedent drug, it's in orange, and the response rate of this new FDA approved drug is in blue. In several cases, you can see that the orange bar is longer than the blue bar. That means the response rate that you could already have with the existing drug was in fact higher than the response rate that was available with this new drug that was just approved on the basis of response rate itself. That's why without a comparison, without a concurrent comparison, it is very difficult to say whether or not the response rate that you are seeing in that single arm trial, is it better than what you already have? Is it worse than what's the best possible case scenario? Without a control arm, it's very difficult to say. And as this study shows, in most of the cases, drugs that were approved on the basis of single arm trials, they actually had a response that was poorer than what already existed. With regards to sample size, this was again a very interesting study that looked at drugs that were approved on the basis of single arm trials and looked at what the sample size would have been needed to run a randomized trial in the same setting. And they saw that theoretically it would have been possible to do randomized trials within the comparable duration for the trial to be completed for more than 80% of these approvals with an endpoint of response rate or PFS or OS, even if you used overall survival as the endpoint for a randomized phase 3 trial, in 80% of those cases, you could have actually done that rather than rely on response rate from a single arm trials. If you relied on PFS, you could have done a randomized trial in 95% of the cases. Next, we return to the question of what is an impressive response rate and duration of response. How good is good enough? Here I'll give you an example of FDA approval of nivolumab for hepatocellular cancer, which was based on response rate, and it was based on a response rate of 14.3. So FDA approved nivolumab for hepatocellular cancer, second line, based on response rate from a single arm trial, and that response rate was only 14%. Only 14%. However, I'll give you an example of this sorafenib trial in desmoid tumor. And this was sorafenib versus placebo. Here, the response rate in the placebo arm was 20%. 20% of the patients in the placebo arm had an objective response. So our nivolumab in hepatocellular cancer was approved on the basis of a response rate lower than that of placebo, albeit in a different tumor. But the point I'm trying to show here is we do not have a good definition of what is a good response rate. And sometimes we see that even patients who are getting placebo can respond. So 14% is, I think, we should all agree that it is too low a response rate for any drug to receive approval. And not surprisingly, nivolumab's approval got withdrawn in this indication based on results from a randomized trial. And in fact, this is not a new phenomenon. We had studied this for targeted drugs for both response rate and duration of response. And we commonly see that the response rate and especially duration of response are very much inflated in non-randomized single-arm trials compared to randomized trials. That means the same drug in the same setting, you see an impressive response rate and duration in a single-arm trial. But if you do the same, if you trial the same drug in a randomized fashion, you see that the response rate and duration usually fall. As shown in this figure, we found that the duration of response is usually 17% higher in single-arm trials for the same drug indication as compared to randomized trials. Next is the question of what is rare. And there are definitions of how to define rare. In Europe, we define it as less than 6 per 100,000 people incidence rate. But, nowadays, because we are redefining all the tumor types based on specific molecular subgroups, any tumor can be subdivided into several rare tumors. If we look at non-small cell lung cancer, for example, then we see that non-small cell lung cancer, which is the commonest cancer in the world, is actually a combination of several rarer subgroup of cancers. So in theory, we can claim lung cancer as rare cancers based on specific molecular signatures. And similarly, what is an unmet medical need? We say that single-arm trials might be enough when there is an unmet medical need, but what does it actually mean? This study looked into it in a little detail, and they found that even when the trials were citing unmet medical need, the incidence of the cancer in the US quite varied. Yes, there were some cancers that was pretty rare and might have signified unmet medical need, but there were also cancers that were quite common. And in fact, they also looked at availability of treatments in the NCCN guidelines and the 5-year survival rates. And they found that even though they were citing unmet medical need, in several of these occasions, there were several alternate treatments that were available in the guidelines, and the 5-year survival rates were pretty high. So unmet medical need is probably not always truly an unmet medical need. And then there is the issue of assessing safety from single-arm trials, which we know is inadequate. And actually the regulators know about this, because in this perspective, the members of the FDA published in New England Journal of Medicine in the context of Keynote 183 and Keynote 185 trials in multiple myeloma, which were halted prematurely due to safety signals. They clearly mention here that although many cancer drugs have been approved on the basis of durable response rate in single-arm trials, it's difficult to adequately evaluate efficacy and safety of drug combinations without a control group. And overall survival can be accurately assessed only in a randomized trials. So they do caution about single-arm trials and the fact that safety cannot be adequately assessed. And in a randomized trial only, it can show up as increase in hazard ratio for overall survival. Finally, let's discuss about the societal impact of the drugs approved based on single-arm trials. In this analysis that we published in BMJ, we looked at drugs that received accelerated approval and then ultimately failed in the confirmatory trials. We found that majority of them were actually drugs that received initial approval based on response rate, 15 of the 18 trials. And if we look at drugs that had to be revoked or withdrawn, we see that most of them were actually initially based on response rate, and they stayed on market for a substantial number of years before getting withdrawn from the market because they failed in the randomized trials. And further, if we look at what's happening all over the world, this was an important survey that was done by the WHO EML group that surveyed oncologists practicing in different parts of the world and asked what are the top 20 cancer drugs that they would consider absolutely essential. And if you look at the list here, you find that none of the drugs that they mention here, be it from high-income country or low-income country or middle-income country, none of these actually are drugs based on single-arm trials. So in terms of the societal value of drugs that received approval based on single-arm trials, it shows that it's probably negligible. So towards the end, let's discuss when can these single arm trials be useful and can be used. So the way forward, in my opinion, is to use response rates from single arm trials as accelerated or conditional approval, and we should not be giving regular approval. It cannot be justified based on short-term responses from single arm trials alone. Accelerated approval provides the necessary compromise between access and evidence. But the question is, how do we define impressive responses? And I think one way forward would be to measure not just response in single-arm trial, but to measure the difference in response rates in a randomized trial to give initial accelerated approval, and then that same trial can be followed up for survival endpoints to confirm the benefit. And the other idea is actually to measure the durable complete response rates, because complete response is a meaningful endpoint, and complete response is undoubtedly the function of the drug. And it is quite meaningful for the patient as well to be declared as having a complete response, which is not possible without the drug. And the other option is to measure milestone durable response rates, for example, response at 12 months or 9 months. And sometimes in rare cases, as shown in this figure from the Javelin-Merkel 200 trial, overall survival might also be meaningful to look at, especially when the natural history of the disease is very clearly known and is quite poor. For example, without the treatment, if we know that a majority of the patients die within 6 months, and with treatment, we have majority of the patients living up to 5 years, then the benefit is obvious. And because this is the MCBS session, we should also ask this question: does the MCBS scoring need to be adjusted? At the MCBS, we are quite open to adjusting our criteria so as to make it up to date and most relevant. And in fact, we published a paper last year about what are the possible pitfalls in appraisal of MCBS scores and how we can improve that. And as you can see, one of the points that we show here is single arm trial. As you can see, we do recognize single arm trial as one of the possible pitfalls here, but how can we improve it? That's open for discussion. So this is the MCBS form for scoring based on single arm trial and response rate. But if you look at this form, I think there are a couple of points that we should think about carefully in the future. One is that we allow scoring on the basis of PFS as well from single-arm trials. And as we have discussed, PFS is not a reliable endpoint for single-arm trial because it could also be a function of the natural history of the disease. And we cannot definitely say that PFS is from the drug activity. So that PFS, this is a little counterintuitive, but response rate is the best endpoint for a single-arm trial and not PFS or even OS. And the other thing is we provide an upgrade based on phase 4 experience, and I think we need to be a little more explicit about it. Rather than phase 4 experience, maybe we should also consider differences in response rates if a randomized trial is conducted subsequently. So in conclusion, single-arm trials are not meant to provide evidence sufficient for approval. However, that ship has sailed and we have several drugs that are approved on the basis of single-arm trials, but we need to make sure that those approvals are accelerated or conditional approvals, not regular approval. We should not allow premature regular approval based on single-arm trial, because once a drug gets a conditional approval, access is not an issue. Patients will have access to the drug anyway, but we should ensure that robust evidence follows. Long-term follow-up data are needed to develop confidence on the efficacy outcomes that are seen in single-arm trials. And in many cases, single-arm trials are already large and long enough that we could actually have done an RCT. And we should be aware of several biases in evaluating overall response rates from single-arm trials, as we have seen that the response rate and duration of response can be falsely exaggerated in single-arm trials compared to RCTs. I'd like to thank the ESMO committee for inviting me for this talk. I'd like to thank Professor Cherny, Professor Elizabeth DeVries for their continuous support and encouragement, and for allowing me to work with the wonderful ESMO MCBS working group members. And a special note of thanks to Laura Carson from Queen's Global Oncology Program for helping me with the slides. Thank you. I'm happy to take any questions.